Dr. Manas Mandal is an associate professor at Roseman's College of Pharmacy. In 2019, he was awarded a Fulbright Specialist appointment by the U.S. Department of State's Bureau of Education and Cultural Affairs and World Learning. His expertise is teaching and research in immunology. He received a PhD in science from the Jadavpur University in India and did postdoctoral research at the University of Chicago and the University of Michigan. He was also involved in biodefense research at Virginia Tech. Dr. Mandal has given invited talks in the US and abroad and chaired scientific sessions. He has mentored many students, fellows, and faculty, and is professionally associated with numerous pharmacy education, research, and accrediting organizations. Dr. Mandal is also a member of the American Association of Immunologists and the American Association for the Advancement of Science. And now, it is our pleasure to hear from Dr. Manas Mandal.
Thanks, Brenda. Thank you very much for your kind introduction. Hello, everybody, and good afternoon to the audiences in the United States, and I believe I have some audiences in India. Good morning to them. It's across the world. It's a new day there. Let's talk about COVID-19. We are living in this pandemic for about 6 months now. When it was all beginning, as an immunologist, I wanted to understand this disease, how it impacts us, what are the pathophysiologies, how we respond immunologically as a host to the infection. Subsequent to those informations, we will look into the detections, assays for this infection and attempted treatments. I will end up today's talk discussing various vaccine development approaches currently pursued at various levels throughout the world. At the very beginning of this presentation, I would like to acknowledge various SARS-CoV researchers and clinicians who are fighting this disease heroically. And many of, if not all of this scientific documentation that has been used in my talk are freely available. This is T.S. Eliot in 1925, aftermath of World War I. I would like you to take a pause and think about it. What is gonna be? Ending with a bang or with a whimper? I will answer that question at the very end of my talk. At the very beginning, meaning this is how it started. On January 9, 2020, Chinese researcher, scientist, Dr. Zhu Jiangguo, identified a pathogen in an individual apparently suffering from pneumonia in Wuhan province of China. Subsequently, that was designated as novel coronavirus, and currently we know it as SARS-CoV-2. In between, from that point up till now, this has been the most talked, debated, telecasted, and noted events all over the world, perhaps more so than landing on the moon by a human being long time back. As I was preparing this lecture, these slides, I realized that I have included a lot many information that perhaps I could accommodate within the allotted time of about 45 minutes. So what I'm gonna do today is not reading the slides, rather giving you the main concepts regarding the pathophysiology, infections, detection, treatment, and vaccine development approaches. If you want to go in detail, you will have the access to the materials that will be posted later on by the university. So let's understand a little bit about virus infection. Viruses are obligatory intracellular microorganisms. They live inside the host cell and where they replicate, meaning viruses are inert particles. They would either contain RNA or DNA, but not both. Therefore, they need to enter into a cell type, meaning host cell, to trigger the infection. Upon infection of the host cell, infected cell dies due to direct cytopathic effect of the viruses and lysis of the infected cells. Who lyses them? Some of the white blood cells that we all carry in our circulation, such as cytotoxic T lymphocytes, CTL, CD8 positive T cells, or natural killer cells, NK cells. These are the two cell types that are entrusted with clearing up the virus infection from our system for any virus infections and fighting cancerous development in us. Subsequently, the Coronaviridae Study Group, CSG, is a worldwide group that determines the nomenclature of any viruses that we know of today. Current virus COVID-19 belongs to severe acute respiratory syndrome coronaviruses SARS-CoV and designated it as severe acute respiratory syndrome coronavirus disease, SARS-CoV-2, or in short, COVID-19. This is the timeline. What it shows here that we had coronavirus infection in the world many years prior to the current SARS-CoV-2 infection. I do not have all the details, meaning it goes back to 1960s, but in recent years, in 2003, we had the severe acute respiratory syndrome virus infection. Particularly, it impacted Far East in Hong Kong areas. Subsequent to that, we had Middle East respiratory syndrome, MERS, that impacted Middle East in 2012. Current infection is noted as SARS-CoV-2. It's the same family of coronavirus infection triggering coronavirus disease 2019, COVID-19. Here's the structure, overall structure of coronaviruses. They belong to the family of Coronaviridae, and these are the family members, SARS-CoV, MERS-CoV, and SARS-CoV-2, the current infection. It's a single-stranded enveloped virus, have distinct club-shaped spike protein, S protein that you see here. And if you can imagine, those spike proteins are radiating out of sun, solar corona, that's gotten them the name of a coronavirus. Thus, so far, 14 mutations in the S protein has been found. One is of particular importance in the context of its pathogenesis and infectivity. Perhaps D614G mutation in S protein was originated in China or in Europe, but currently it's distributed in US also. So this is the overall structure of a coronavirus that contains on its surface spike glycoprotein S protein, membrane protein M protein, and nucleoprotein. These are the structural proteins and genomic RNA that this virus contains. Along with that, we have enveloped small membrane protein and hemagglutinin esterase that actually utilized by the virus to cleave off budding viral particle from an infected cells, then released it to infect the adjoining subsequent cells. Few words about coronavirus infection, current infection, SARS-CoV-2, and why it is so dangerous. Because essentially, it jumped species. It was not originally found or co-evolved within human population. Most of the other virus infection that we have today, they co-evolved with us, meaning we have them for centuries or generations. When that happens, viruses learn to live in its host. As I mentioned before, that because they're inert particle, so it's in their interest, they got to keep the host alive. Yeah, it's funny, but that is true. And the population, meaning the host, human being, survives with much of illness from that virus infection. But that is not the case for this current SARS-CoV-2 infection. It jumps species. Technically, that is known as zoonosis. Where it was found, usually it is found in bats and could have jumped to humans via an intermediary such as civet cats. Little bit about infectivity and mortality, since I started with saying that this is a dangerous infection. And these are some of the numbers. Now, these numbers are not very hard and fast number, meaning they will be varying from region to region. And as we move forward throughout the world in the context of this virus infection. So these are not hard and fast number, but these are this infection is highly transmissible, increases vulnerability of infection and diseases. High mortality rate with infection fatality ratio has been calculated to be 0.65% currently by CDC. And estimated basic reproductive ratio R naught value varies very widely, commonly ranges between 2.2, 3.9. In comparison to this R naught value for COVID-19, if I can bring a parallelism, we all know of Spanish flu. By the way, that was not originated in Spain. That was perhaps originated in Texas or Kansas City in US, but it was named Spanish flu. That Spanish flu infection R naught value is around 1.4 to 2.2. So you can imagine how more infective this virus is. This R naught value refers to how many other individuals can get infected from one individual. The first infected individual is represented by R naught, and how many other individuals can get infection if they come in contact with that first infected individual. That is defined as the reproductive ratio or R naught value. Overall estimates of mortality, that also varies. Currently estimated to be 0.5 to 15%. That is a high mortality rate, and particularly persons aged over 65 getting impacted, meaning they are dying in huge number. Prevalence of asymptomatic infection, that also varies 20 to 45%. In US, current estimate is about 40% of the individuals are asymptomatic. But asymptomatic people can also transmit the infection, and there it becomes more critical or more difficult to manage, and that transmissibility can stay longer than 14 days. About half of the COVID-19 transmission occurs even before one feels sickness, meaning if I am currently carrying COVID-19 virus in me, I may be even asymptomatic, and thus I may transmit the virus infection as I'm talking to people, as I'm mingling with people, even including my family members. There comes the logic of physical separation and mask wearing that breaks the chain of transmission, and we will examine those issues at the very end. Let's look at the human receptors that are utilized by this virus to gain access into us, meaning setting up of the infection. There are ACE2 receptor structures that has been described heavily in the literature that are involved in original SARS-CoV infection and current SARS-CoV-2 infection. And this receptor structure is also found in animals such as bat. Also, there is another receptor structure that are involved in priming of this viral entry, followed by binding to S2 receptor structure is TMPRSS2. This molecule primes a structural aspect of the virus, viral S protein, that allows it to gain entry into the cells. And that is the information shown in this slide and the next one. Without going to the detail, it is sufficient to say that S2 and TMPRSS2 expression is found in all over in our body. Particularly, they are overexpressed in nasal epithelial goblet cells, ciliated cells, nose, alveolar structure in the lung, gut, and heart tissues. And the skewed expression of these viral receptor structures that dictate the entry associated protein in the upper airway may be correlated with its enhanced transmissibility. Meaning, one infected individual, when it sneezes or talks, there is always biological particle, fluid particle that goes out of their mouth, and usually that lands in front of our face, and we inhale them, and if it lands in our body, then we touch our hands. That leads to the transmission of the infection, and it's a mucosal infection in that sense. Little bit of structural aspects of this SARS-CoV spike glycoprotein or S protein. It's a trimeric structure. It has S1, S2, and S3 subunit. S1 contains the receptor binding domain, so-called RBD, and that directly bind to the peptidase domain of this S2 receptor. Whereas S2 is responsible for membrane fusion, meaning a viral particle first needs to bind, then fuse with the host cell membrane so that the viral particle gains access inside the host cell, where they will hijack the protein synthesis machinery of the host cell and dictate them to make viral protein and peptides. and new viral particles will be expressed on the infected cell surface that will be helpful in spreading the infection within that same host or releasing that viral particle through our talking, sneezing, etc., so that subsequent someone else can get infected. And if you want to look into the detail, you can read this information at your own pace. But here is a notable information. SARS-CoV-2 protein binds to this peptidase domain of H2 receptor structure with high strong binding that is described as a dissociation constant scientifically, which is about 15 nanomolar concentration, which is a very, very high strength binding. So with this high strength, this virus gains entry via H2 in our body and sets up its shock or infection. And down below, this shows that how this viral particle spike protein is bound by the H2 structure. And this is the science article. These are all freely available. Although H2 is hijacked by the coronaviruses, its primary physiological role is not this attachment to the viral particle, rather maturation of angiotensin, which is a peptide hormone that controls the vasoconstriction, blood pressure. Therefore, H2 decreased expression associated with cardiovascular disease, and my pharmacist students and colleagues know that cardiovascular disease due to H2 decreased expression can be treated with benazepril or lisinopril type of medications. And these are the medications used in heart failure and prevention of the strokes. This is little detail. I'll gloss over this information. Now, let's come to the clinical presentation. So after the virus gains entry in our body, particularly in the upper respiratory tract, nasal mucosal passages, then it flows through our gut, flows through our nasal passages and enter into our gut. And what type of pathophysiological manifestations it presents with. Typically, there will be fever, dry cough, exhaustion, anorexia, smell and taste disorders, overall myalgia, and shortness of breath. Less frequently, we can see nausea, diarrhea, sore throat, headache, and cutaneous manifestations such as in the toes that you see up here. Those are the mild manifestations. More severe presentations includes neurological complications, acute respiratory distress syndrome, ARDS, that is the most dangerous and complicated implication of this virus infection. I recently described multi-system inflammatory disease in children, particularly. We simply do not understand why specifically in children. How it generates or triggered, we will look into some of the data later on. Cardiac, it's known to trigger cardiac injury, acute kidney injury, liver dysfunction, thrombotic complications, meaning clotting cascade defects, shock and multi-organ failure. That's particularly compromises the life. And many times there will be bacterial co-infection, because if the lung is engorged with the mucus, that is a fertile ground for developing secondary bacterial co-infection. Comorbidities associated with this infection, particularly with severe presentations of the infection, cardiovascular disease, diabetes, hypertension, chronic lung illness such as COPD, and kidney disease. And on the right-hand side top panel, you see the infected individual toe, swollen toe with part place, meaning there is lack of oxygen, and this is due to the COVID-19 infection. And mid panel shows the engorged lung and this glass-like opacity. That is one of the hallmark of severe infection with COVID-19. Bottom panel examines white blood cell infiltration in the alveolar tissue. Usually in a normal individual's lung alveolar tissue, if we do a histopathology, we would not have these many neutrophil monocyte and alveolar macrophage infiltration in the lung. But in this infected individual, you can see the lung is heavily infiltrated with these white blood cells. And ultimately, overactivation of these white blood cells towards this infection will trigger the major immunopathophysiology of this infection. Before we discuss the immunopathophysiology and pathophysiology of this discussion, Let's take a look at the time course that will help us understanding the pathophysiology. The infection starts very early on in an individual. Early on meaning if day 0 is the onset of the symptoms of the infection beginning with a fever, infection may have gotten into that person at least 5 days before. So -5 to -10 day is the incubation period with the virus. At day 0, we might see the symptoms of infection such as fever. Peak transmissibility of this virus infection is about -half a day, meaning about a day before first sign of infection such as fever, even if it's mild, that is the peak transmissibility period. Then this viral load can be tracked in nasopharyngeal swab, peaks around day, I would say 5 through 7, then it goes down. Around day 3, we will have the IgG class of antibodies, first beginning with IgM class of antibodies, subsequently IgMs will be converted into IgG class of antibody, and that will reach a plateau about day 12 of the infection. So antibodies can be detected to establish the infection or ongoing or previous infection within first 3 to 5 days upon infection. And this is the window of pneumonia and, if necessary, mechanical ventilation. With that schematic infection diagram, let's look into what exactly it does. Before we do that, let's first quickly glance through immune responses to virus infection. Usually, we trigger innate and adaptive immunity to clear up the virus infection. Yes, antiviral drugs do not clear virus infection. Antiviral drugs only inhibits viral replication. That's true for all virus infections in us, including this one. Our white blood cells, that ultimately clears up the virus infection. What are these white blood cells? As I mentioned before, natural killer cells, part of innate immunity, whereas as part of adaptive immunity, cytotoxic T lymphocytes, these are the 2 cell types that will engage in an infected virus-infected cells and kill them, releasing cytotoxic chemicals and granules that will clear up the virus infection reservoir from our body. Whereas neutralizing antibody that you may have heard so many times, they are also protective. When they can protect, as long the viral particle is not growing inside the cell, rather than it's in between tissue spaces, because antibody is a Y-shaped structure. It cannot visualize what is growing inside the cell. Whereas when the viral particle is in between tissue spaces, antibody can certainly bind to it and clear up the infection through various mechanisms such as receptor-mediated endocytosis, phagocytosis, and clearances by phagocytes, complement activation, etc. At the very beginning of the infection, what protects us or trying to protect us against any virus infection, including this one, type 1 interferon response, such as interferon alpha and beta. What has been observed, as I will show you subsequently, that in many of the severely diseased patients, this type 1 interferon response is compromised, not adequate. That leads to the severity of the infection. And here is the immunopathophysiology of COVID-2 infection in lung. Without going into this detailed diagrammatic representation, in a normal lung, this is the scenario. We have only very few alveolar macrophages, capillary is wide open, etc. It's not engorged. Alveoli is sufficient, means it has sufficient space for exchange of oxygen, meaning we have regular flow of oxygen throughout our tissue, normal lung function. Whereas in SARS-CoV-2 infection, you can see these alveoli is the smallest unit of our lung. It's engorged with many white blood cells, dead tissues, dying cells that may limit the gas exchange, meaning compromising oxygen exchange in our lung, therefore lack of breath, respiratory distress. How it happened? Various immune cells such as T cells, NK cells, neutrophils, and inflammatory monocytes particularly are rushing into this alveolar structure in the infected patient's lung. Out of the capillary circulation, you can see this white blood cell exiting out of the capillary blood vessels that supplying our lung and reaching to the infected lung tissues. Thus, at that point, they are releasing inflammatory cytokines such as IL-1 beta, interleukin-6, interferon gamma, and TNF-alpha. These are the inflammatory cytokine. They constitute so-called inflammatory cytokine storm, leads to the severity of the infection. And what I was mentioning prior, that this opacity, glass-like opacity, this is the electron micrograph and CT scan of a 3D modeling of a CT scan of a 59-year-old individual who eventually died at George Washington University. And you can see the scarring of the lung, this yellowish, greenish yellowish areas, and the glass-like opacity that you see here. That's how the lung presents of an individual deceased due to this infection. And right-hand side panel shows the electron microscopy of the purple cell type infected with so many viral particles isolated also from a patient sample. So what are the regular clinical features of COVID-19 infection? These are predominant clinical features. Older males, above 50 years old, likely to develop severe COVID-19 infection. The most common clinical manifestations at the onset of illness includes, as I mentioned, fever, cough, fatigue, myalgia. Severe cases may frequently would show dyspnea, distress breathing. In more severe cases, acute respiratory distress syndrome. And with that, we will have leukocytosis, meaning lower white blood cell counts, lymphopenia, lowering of lymphocytes, more common in severe cases than in moderate cases. That leads to the clue that severity of the disease depends on how our white blood cells respond to this infection. As a biomarker in the plasma, we can track ALT, LDH, lactodehydrogenase D-dimer, various inflammatory markers such as C-reactive protein, ferritin. Those will be present at a high level in severe cases compared to the moderate disease condition. And these are the inflammatory cytokines that will constitute the cytokine storm. Also, we will observe type 1 interferon alpha and beta impaired response that will lead to the disease severity. An underlying pathophysiology of pneumonia is elevated inflammatory cytokine release in the blood of the hospitalized patients. And this inflammatory cytokine will be released by white blood cells that will be recruited to the site of infection, particularly in the lung, and lung will be filled with mucus and dead cells that will clog up the lung. Some COVID patients recover sometimes with no more support than oxygen breathed through nasal prongs intubation, but others deteriorate, often quite suddenly developing acute respiratory distress syndrome, oxygen level plummets, struggle even harder to breathe. And we have observed the X-ray computed tomography showing the lungs are riddled with white glass-like opacities in last couple of slides and end up in ventilators, and many die. That's where it ends. Little more on white blood cells immunopathophysiology. We have observed lymphopenia, reduction in lymphocyte counts, particularly CD8 and CD4 T cell compartment, particularly in CD4 T cell compartment, CD4 T cell exhaustion, meaning they just can't keep up with the virus infection, means they lost their fight against this infection. That is technically known as CD4 T cell exhaustion. The CD4 T cell exhaustion is one of the hallmark of immunopathophysiology of COVID-19 infection. But in contrast, it has been observed that B cell proportions and numbers are not reduced, which is not a bad news, because if B cells are intact and they can respond to this infection in a proper fashion, proper manner, then at least we will have antibody responses, and hopefully those antibodies will be neutralizing type of antibody due to infection or vaccination. And majority of the patients exhibited a broad significant decline in all the lymphocyte subsets, excluding the B cells. And also we have mentioned about the impaired interferon responses such as type 1 interferons. Now, there has been loss of smell, myocardial injury has been described in the literature due to various facts such as viral invasion also attacks non-neuronal cell in the nasal cavity, olfactory epithelium, and in the brain's olfactory bulb, all expresses virus capturing receptor structure. Thus, direct or indirect myocardial injury or stress is likely to be caused by direct SARS-CoV-2 myocardial infection or due to a cytokine storm or hypoxia. Particularly, very recent publication in JAMA Cardiology documented that heart damage in nearly 20% of the patients out of 400 hospitalized in COVID-19 patients in Wuhan, China, whereas in another study, 44% of 36 patients admitted to ICU had arrhythmias. We have also observed blood clots in many COVID-19 patients, such as 38% of the patient had abnormal blood clotting in a Dutch ICU, and clots from the arteries can lodge in the brain and causing strokes. And ischemia in the fingers and toes can lead to the swollen painful digits that has been observed like this. And so what is the summary of the clinical presentation? Estimated incubation period is up to 14 days from the time of exposure. Median is about 4 to 5 days. Spectrum of illness can range from asymptomatic infection to severe pneumonia, ARDS, and death. In China, about 81% of the cases were mild, 14% severe, 5% critical. Whereas in US, with this data set, 86% has cough, 85% chills, and etc. Common lab findings: leukopenia, lymphopenia, impaired type 1 interferon responses, inflammatory cytokine storm, elevated aminotransferase level, C-reactive protein, D-dimer, ferritin, LDH levels are high. Patients at highest risk at any age with underlying following conditions such as hypertension, cardiovascular disease, elevated lipids, diabetes, chronic respiratory disease, cancer, renal disease, and obesity. Let's switch the gear. How do we detect COVID-19? 3 distinct test platforms finally hit the market after long hiccups in the very beginning. First, RNA detection via real-time RT-PCR that will detect early disease or infection in patient population and can be also used for contact tracing. It takes some time to get the test results. It varies currently between few days through weeks in different parts of the world, including US. Antibody detection test via enzyme-linked immunosorbent assay, ELISA. Antibodies comes at a later phase of infection, but as early as day 3 to 5 of the infection, we can detect antibodies beginning with IgM, then IgG class of antibody. These tests can be used for surveillance and monitoring purposes. Finally, very recently released and developed test is antigen test for rapid detection of the virus. These are immunofluorescence-based lateral flow technology that is a qualitative detection for SARS-CoV-2 antigen, particularly targeting spike protein antigens. And I will not go into the details of the individual diagnostic tests in the interest of time, but you can read at your time. What is the intended use for RT-PCR? Intended use clinically detect infection in an individual, but negative results do not preclude COVID-19 and should not be used as sole basis for patient management decision. 2 consecutive negative test results within 24 hours should determine no infection, and negative test results must be combined with other clinical observations such as patient history, where you have been, other epidemiological infections. And that is known as serological tests, antibody tests that I talked about before. And when antibody is detected through this ELISA test, which is known as serological tests, that does not determine the protective immunity due to presence of antibody, meaning how much antibody is protected or duration of the protection or immune correlates of the protection. None of these informations can be obtained simply doing an antibody test. Actually, immune correlates of protection or duration of the antibody circulating in the patient's plasma or recovered individual's plasma, how long it will be remain protective is a $1 million question. And it varies from every infection to infection. To understand and determine immune correlates of protection, it is not an easy task to do that. Finally, rapid antigen testing for SARS antigen. This is the immunofluorescence-based assay, currently very popular. This is a field test that can, within 15 minutes, can give a yay or no response through nasopharyngeal or nasal swab sample out of a patient or suspected infected individual. But there is a caveat. Up to 15% of the false positive reports has been observed with the SOFIA or rapid antigen test. We have to be careful about that. Next, let's take a look at the treatment options considered for COVID-19. A panel of US physicians, statisticians, and other healthcare experts developed a treatment guideline for COVID-19 because it is so new, there is no standard textbook recommendations yet. Therefore, the following drugs are a recommendation for the use of treatment of COVID-19. Rather, they should have been used or should be used in a clinical trial setup. These drugs comes in the category of repurposed drugs, such as HIV protease inhibitors, type 1 interferon, hydroxychloroquine, azithromycin, steroids, interleukin-6 inhibitors, JAK inhibitors. And why these are repurposed drugs? Because these drugs have been developed to treat a different disease condition and gained FDA approval now. In an emergency situation like this, these drugs has been clinically trialed in a hospitalized patient so that if they can bring some benefit to the patient population. Many of these are showing promising results, such as interferon treatment, tocilizumab, IL-6 inhibitor antibody drug, and anti-malarial drug has been discarded, not doing anything, rather cautioned against it. We'll go through some of those drugs individually. There are other category of drugs, such as investigational drugs, antiviral, such as remdesivir, one of the most touted antiviral drug that has been clinical trial. There are some positive data about this drug, and favipiravir, another general purpose antiviral drug that is being also clinically trialed. Convalescent plasma, we have all talked about. Essentially, these are anti-CoV-2 antibody directed towards spike protein, and they have been now developed in the lab setup as a monoclonal antibody drug going through phase 2 and 3 clinical trials. They should come out in near future. Various concomitant drugs such as RAS inhibitors, ACE, ARBs, such as these drugs, non-steroidal anti-inflammatory drugs, ibuprofen. Very recently, steroids such as dexamethasone shown improvement. And these are some of the information on ACEs and ARBs. Cardiac patients managing their hypertension, cardiovascular diseases depends on the drug. There was a concern because these drugs are involved in increase the expression of H2 receptor structures that are involved in binding of this virus. But clinically, it has been observed that ACE inhibitor or ARBs in the patient population with hypertension and hospitalized for COVID-19 was associated with similar or lower mortality compared to patients who are not taking those drugs prior to the infection. Other studies suggest that use of an ACE inhibitor or ARB is not associated with an increased risk of acquiring SARS-CoV-2. So that's a good news. NSAIDs such as ibuprofen that can be used to reduce the fever, headache, and etc. And there is no convincing evidence that NSAIDs are especially dangerous for the patients with COVID-19. And sometimes NSAID or an acetaminophen can be used when used for continual fever suppression may reduce the immune responses, white blood cell response towards the virus infection, because these are anti-inflammatory drugs and immune response begins with controlled inflammation. So there is other aspect of continued use of those drugs. I better don't talk about this, right? But let's say this. Chloroquine and hydroxychloroquine has no medical value benefit in treatment of COVID-19 patients. NIH specifically recommends against it. FDA has cautioned against use of hydroxychloroquine because it triggers QT prolongation, ventricular arrhythmia, and sudden death syndrome. Recently, corticosteroid clinical trial, dexamethasone, has been done in UK in conjunction with Oxford University and shown some promising result in controlling severe inflammation. Study indicate that steroid did not significantly reduce the risk of death. In totality, 21% who received dexamethasone and 24% patients allocated to casual care died within 28 days. But statistically, one can draw a conclusion this is slightly significant statistically. But corticosteroid use use also increase the duration of hospitalization through 10 days approximately with prolonged viremia, meaning steroid being a global anti-inflammatory drug, when used into the patient population, that may increase viremia, meaning viral load in the patient population, thus increasing their hospitalization, hospital stay, but certainly it can control inflammation. Remdesivir is an investigational broad spectrum antiviral drug, but needed to be given IV in hospital setup. Thus, it has limited usage. This is the clinical data. 61 hospitalized patients, 57 on ventilation, treated with remdesivir, 68% shows improvement in terms of oxygen support. And also, some benefits in severely ill patients has been observed. Reduction with a faster time to recovery, 11-day in the treated patient population compared to 15-day in the placebo group. Also improved mortality rates, 8% versus 11.6% in the control group. So this is a drug that can bring some level of relief in the patient population, but it needs to be further researched. Convalescent plasma, such as monoclonal antibody, Eli Lilly, Regeneron, they are all going full steam developing various monoclonal antibody therapeutics and treatment, and clinical data shows very promising results neutralizing the viral load in the patients treated with this monoclonal antibody. These B cells has been isolated from an infected individual. Then in the lab setup, they clone the B cell genes to develop this monoclonal antibody, and that becomes the drug. So therefore, prior to arrival of a vaccine candidate in the market, this antibody therapy might be beneficial for prevention of the infection in healthcare workers, family members of the infected individuals. Yes, mask prevents transmission and infection. Here is the data. Without going to the detail, this Duke University researchers very recently, very recently, this data is about 3-day old, came out in Science Advance rapid publication. Fitted N95 mask prevents its almost highest level, followed by surgical polyester, polyethylene cotton mask. In summary, putting up a mask, face mask, prevents transmission, and this is the relative droplet counts prevented by these individual masks, and this is the relative stay of the droplets in the ambient environment where someone can get exposed without using a mask. And the green, no mask, whereas red is bandana wrapped around the face. That actually doesn't do any good, but all the other first 5 masks, you do not see a transmission. They are baseline data. The last few minutes, I will focus on COVID-19 vaccine development approaches, and worldwide at this point, perhaps upwards of 140 different vaccine formulations being tried worldwide, different laboratories to develop vaccine candidates against COVID-19. People are working almost day and night on a world footing basis to develop COVID-19 vaccines. And this color diagram shows protein-based subunit vaccines are the leading vaccine candidate, followed by the viral vector containing mRNA or DNA containing vaccines. They are high in number. And this is the schematic diagram that shows how these various vaccines will be triggering immune responses. And these are the technologies that has been adopted by various researchers, such as you have heard about Johnson & Johnson, Moderna, and Oxford universities. They are utilizing replication deficient adenoviral vector to ensure the mRNA sequence out of SARS-CoV to trigger immune responses. And people are using pox virus as a vector, whereas protein-based vaccines are utilizing particularly S protein. Many are utilizing virus-like particular vaccine. And one notable vaccine candidate that is in development in India, I believe it's in phase 2 clinical trial in India, Covaxin, being developed by Bharat Biotech in conjunction with Indian Medical Research Council, ICMR, Indian Council of Medical Research in India, touted to be released soon. These are one of the major prominent vaccine candidates. By no means I am advocating for one versus the other, but I myself worked many years as a viral vaccine researcher. Thus, it is kind of dear and close to my heart, and that made me follow this story of vaccine development. These are the various vaccine candidates that will be soon hitting the market. I will end up my presentation in a few minutes talking a little bit about ethics in COVID-19 patient treatment. This is an emerging field, yet it's a gray area. What should we do when there are 10 ventilators available and I have 20 patients needing ventilators? Or I can only enroll 10 individuals in remdesivir clinical trial protocol, whereas there are 50 people waiting in the line. They can be benefited through remdesivir treatment. So clinicians, scientists, citizens, informed individuals should all come together to develop a conversation. How do we address these issues? Because these issues have not been observed. We never faced this situation before. And more so when vaccines become available, who will have the priority to immunization? But at the end, this is my favorite quote. Many of you have seen this quote from me before. It ends, these knew what it was from their experience and had no fear for themselves. For the same man was never attacked twice, never at least fatally. This is the description of plague, 430 BC. This gives me hope. There are many unknowns, but we have learned much in the last 6 months or so. We have learned that neutralizing antibody does decrease. There is a challenge how to maintain the neutralizing antibody even after vaccination for a long period of time through memory B cell responses, memory T cell responses. We have learned that we can break the chain of viral transmission simply wearing a mask, facility closure, physical distancing. We have discovered pre-existing immunity. T cell responses have been observed in 20 to 50% of the individual who never exposed to this virus infection. Where they came from? Because we have been exposed to other cold coronavirus infections, and all these coronaviruses shared antigenic similarity. Very recently, people have started thinking BCG vaccination may confer protection. That has been observed in the countries with strong correlation reduction in mortality who regularly receive BCG vaccination, such as in India, many parts of the European countries. So early testing, test results, better comprehensive treatment guide, vaccine, antibody treatment will make this infection as a manageable virus infection that may remain in the community for a few years to come. What necessary for this is a central and uniform public health policy. At the end, what gives me hope is tremendous public awareness, dedicated group of scientists, clinicians, and frontline healthcare workers, improved testing, contact tracing, isolation, quarantine, comprehensive public health policy, investments in public health, and changes in public health, national, international collaboration in public health policies that will guide us through this dire time, developing treatment, vaccine development, and vaccination approaches. So I will end my talk going back to one of the early slides. Are we gonna end human civilization going to be ending with a bang, or we go down meekly? I will say that we will come back with a bang, because there are dedicated scientists, clinicians, and frontline healthcare workers. All are working, all are trying to understand this infection, trying to come up with a disease treatment plan, and how we can protect ourselves, community, and population worldwide. Thank you for your attention.
Thank you so much, Dr. Mundell. We so appreciate it. That was a fascinating discussion. If there are any questions, please go ahead and reach out now, and Dr. Mundell will be happy to respond. And in the interim, while we're waiting to see if there are any questions, I will thank everyone for their participation. And we'll thank Dr. Mundell for a fantastic presentation. It was wonderful. And I will remind everyone to please watch for future Roadrunner Resource Series presentations. You can find more information on Roseman's website under roseman.edu/alumni. Dr.
Thank
Mundell,
you.
I'd like to answer that.
Okay, so I got the first question, which I'll repeat the question, then try to answer if I know correctly. Disclaimer, I'm not a clinician. Recently, hydroxychloroquine, this is a question from Dr. Chakraborty from India. I know him personally. We are colleagues and we are great friends. Recently, hydroxychloroquine has been reported to be of great help in patient management. The editor of Lancet reported they have been pressurized to suppress its benefit. Yeah, I have actually seen that, and he came up on a webinar. I observed that. It's somewhat political. I know there is a pathway of chloroquine or hydroxychloroquine can be beneficial in this context. Let's talk the science. Hydroxychloroquine for decades has been used to treat autoimmune disease patient populations such as a lupus patient. What it does, it has ability to suppress T cell overactivation that we see in an autoimmune disease patient such as lupus patient. Thus, hydroxychloroquine can act bit of an anti-inflammatory drug or T cell suppression molecule. Also, hydroxychloroquine can eliminate or interfere with the viral particle endosomal fusion, meaning after binding of a cell infected with this virus that is picked up in the endosomal compartment, hydroxychloroquine can interfere in that process. So there is a pathway, scientific pathway, how hydroxychloroquine could work, but it's also very well known that hydroxychloroquine, even to treat a malaria patient, this is the drug that has been originally developed to treat malaria patient populations. There is a monitoring requirement regarding QT prolongation, cardiac involvement. So within that caveat, hydroxychloroquine can be useful, but it should not be taken prophylactically to stop infection in an individual without those monitoring, and only can be given in a clinical trial setup with a qualified physician. Has there been any discussion about do you think that, so second question, do you think that molecules capable of fooling the spike protein by presenting decoy receptors? Yes, people are working. Another question from Dr. Chakraborty. Their structure function analysis has been done already. People are working as a decoy receptor, meaning it's a stereochemistry. If this is the S2 receptor that is binding to the viral S protein antigen spike protein, now if I can block this receptor structure, S2 receptor on my body, on my cell types, with this decoy molecule, steric analog through synthetic chemistry, then when the receptors are blocked, virus S protein cannot be able to or should not be able to bind to that structure. People are working on it. There are animal trials that I am aware of, but I am not aware of any clinical trial moving forward with that idea. But that is a valid idea. The 3D structures and everything has been determined of the S protein and its respective binding domain to the S2 receptor structures. People are working on that aspect. Now, their third question, has there been any discussion about the use of the ivermectin? Sandy, right? There has been a little bit discussion about that, but I'm not very aware of a detailed discussion of the ivermectin type of a medication, so cannot really go into the detail about that. I did not really look detail into it, but to answer your question, yes, there has been some discussion about usage of ivermectin and that type of medication in the context of this infection. But unfortunately, I am not very qualified to talk about that aspect beyond that. Next question is Russian vaccine, Sputnik. Yes, it's Sputnik 5, right? Uh, yeah, they name everything with the Sputnik, with the early success decades back with sending the landing a monkey and a human being on the surface of the moon. But we do not have any clinical information how this vaccine will work other than president's daughter received it. That information I got from the TV interview from Russian President Vladimir Putin. But what type of immune responses it developed, what type of formulation it is. What I got from one of the TV interviews from one of the TV channels is that perhaps they used adenoviral vector also to insert MERS or SARS RNA sequences that will or would trigger antibody responses, but that's all about it. What is the clinical data and literature? They have not released it, but they promised that they will release it soon, but I'm aware of that approach. Proposed mechanism to increase IgG levels after 2 to 3 months when they decrease. Adjuvants. People are using many adjuvants to increase the IgG class of antibody responses. To back up that question, let's look into how we trigger IgG class of antibody responses. It begins with antigen-specific IgM class of antibody responses towards any antigen, be it a protein or carbohydrate antigen. Since any infection, including this infection, will trigger IgM immune responses, antibody responses to begin with within 3 to 5 days, then that will be switched over via cytokine secretion such as interferon gamma response that will switch IgM to IgG. And when we develop IgG type of antibody responses, there is a Th1 type of immune response that can be strengthened, energized through usage of some particular vaccine adjuvants that targets certain innate immune pathways. People are working towards that, and that is a challenge. Once we have a vaccine formulation available and ready to be immunized, how can we increase, not only increase the antibody responses, but increase the duration of immune responses, meaning triggering plasma cell response, plasma B cells, and memory B cells and memory plasma cell responses, and not only enhancing their immune responses, but increasing the duration of memory B cell, memory plasma cell responses specific for perhaps spike protein. People are working on that. That will be the task of appropriate adjuvants, but it's in the works. What are the swab tests detecting to determine positive infection? If someone was unknowingly infected, yeah, no one will get knowingly infected, though it was thought it was just a regular flu, and again, healthy with no symptoms. Is there a test? In that instance, I would say that individual is perhaps asymptomatic individual, maybe got exposed to the viral particle from this virus or seasonal flu, other coronavirus infection, viral particle. Best thing is to do, take nasal swab test, also take a antibody test. Combination of these two tests will tell, yeah, if I have the virus still replicating in me at a detectable level, RT-PCR will detect the mRNA. Whereas if I don't have the viral particle replicating in me, I just maybe got some very, very minor infection, subclinical level, still felt something, I'm not very sure. Most likely there will be an antibody developed in that individual. So combination of those two tests will determine whether this individual got really exposed to the infection, either knowingly or unknowingly. As a bedside RN administering swab for sampling, I have wondered, is there a known benefit to saliva sample versus no, there's, yeah, I'm sorry. Very recent data shows that saliva samples are a better detection source of viral copies of viral particle compared to the nasal swab. But originally, nasal swab just was developed and gained the FDA approval blessings of FDA. That's why deep nasal swab going up in the nasal tract was somewhat painful, discomforting. That's the way it's done. Now, superficial nasal swab is good enough to detect mRNA particles, but very recent data shows that saliva sample is equally important and sensitive for collection of sample. And I can see your concern. As a nurse practitioner, you are in the treatment facility, you are taking care of the patient. Now, if you can give a little cup to the patient and can you drool a little bit? And you can take the sample at a distance and then you take it aseptically and send it to the lab. It's much better than you are almost coming very close proximity to the individual trying to take a nasal swab. Saliva should do that, but you have to follow the guidelines of the commercially available test kits that your hospital or treatment facility is utilizing, whether their test kits can detect saliva-based samples, but they should be able to. That's the scientific information I wanted to give. Is there a truth about the newer mutations not causing as many symptoms? No, it's actually the opposite. The particular mutation in S protein that has been linked to actually severity of the disease conditions, but obviously we do not know, have not learned about all the mutations all over the world and their respective clinical manifestations or clinical involvement in the disease severity. Usually it can go either way. We pay attention to the mutations that are more dangerous, more infective, or makes it more infectious prone, rather than we do not pay attention. Here, with this mutation, this is not causing so much of an infection. So that does not remain very interesting to the scientist or for the clinical purposes. So mutations that are now noted, they are mostly linked with triggering enhanced infection and disease severity. But as molecular biology goes or mutation goes, it can go in either directions. We have to wait until we get much more data that links a particular mutation with a disease severity or less severity. That remains to be seen. But it's a very good question, Ken. Anonymous attendee. There are about different vaccines in clinical trial right now. Yes, that is true. I discussed a few of those. Is it possible we could need more than one vaccine? Yes, that's a great question, actually. Who are a certain age, certain comorbidities. Usually, we never had, usually means so far, we never had a vaccine out of mRNA or DNA sequences other than experimental vaccines went in clinical trials. This is the first time, other than Ebola virus vaccine, we will have a mRNA or DNA-based vaccines. Now, who will get these vaccines? In what frequencies? What are the co or contraindications that needed to be determined? Usually, it has been observed with the live attenuated vaccines, we should not give that live attenuated vaccine, such as adenoviral vaccine is a live attenuated vaccine, to a immunosuppressed, immunodeficient patient populations, pregnant women, or children under 2 years of age. That's the current guideline from CDC in the context of live attenuated vaccine, which will be the adenoviral, replication deficient adenoviral vaccine, kind of falls in category. Whereas protein-based vaccines, no one should have anything to worry about protein-based vaccines, because if they do not trigger immune responses, at least they would not cause any adverse reaction. So anybody can get that. But protein-based vaccines usually needed to be given more than once. Adenoviral vaccines ideally should be once sufficient, but in this unprecedented situation, it may be recommended to give at least 2 doses of adenoviral vaccines, such as Oxford University vaccine formulation or NIAID's vaccine formulation from Moderna lab, perhaps would come out as a recommendation to give 2 vaccine formulation in a span of 1.5 months or so. But it has to be determined case by case basis, who can get it, how many vaccine doses, booster doses, etc. And comorbidities, certain individuals with certain comorbidities, I don't see any comorbidities compromising anyone's ability to receive a vaccination other than live attenuated vaccine formulation. But any protein-based vaccines or killed virus vaccines, such as the Covaxin from India, should be given even patients with higher degree of comorbidity. And actually, patients with comorbidities, they should receive a vaccine because they are the most vulnerable population. Finally, at the outset, thank you, Dr. Malpan. Recently, Russian scientists claim to have discovered weakness of coronavirus through normal water. What is it? I do not understand what is normal water. Is it a particular type of water that is available in Russia? Simply, if you can qualify this question or clarify this question, confirming individual reinfected in a few months after recovery. Yes, individuals can be reinfected after a recovery, but it is hoped that that reinfection after recovery of the primary infection should not be very severe or serious life-threatening condition triggering. That's the hope. But they can certainly be reinfected, and that is true for all diseases, all virus infections, including seasonal influenza infection. One can get reinfected, but that reinfection should not be severe or life-threatening. Why? Because with the first infection or primary infection, that individual would be expected to develop certain level of immune responses that will be protective in nature at a certain level. Since I could not understand that the coronavirus to normal water, I would not be able to understand regular water. No, I have not had such an information that water some way somehow weakens this virus. If that would be true, simply we will be all good. Simply spray water, wash with water, that will clear up any infection from the surfaces. Or if I get exposed, as soon as I suspect such a thing, all I have to do, sprinkle water on my face, means wash my face heavily. No, water doesn't do anything. That's why current disinfectants usually contains at least 10% bleach, household bleach, that kills virus infection, or 60 to 70% ethanol, that kills a virus, particulate matter. Water does not kill a virus. If it did, it will be so easy. I think that's all the questions I do have at this point.
Wonderful. Thank you so much, Dr. Mendel. That was really a fantastic presentation. We're grateful to you for sharing your expertise and your time, and we're grateful to everyone who participated. Thank you, everyone. Again, all attendees will receive Dr. Mendel's slides and the YouTube link in the coming days, and we hope you'll join us for future Roadrunner Resource Series presentations. Thank you all so much, and have a wonderful evening.
Thank you very much. I appreciate it.